Yeah, thanks a bunch for having me here today. It's really great to see so many great presentations until now, maybe. We'll see. So I'm actually wearing two hats today. I'm both with the University of Minnesota, but I'm also with the Minnesota Erosion Control Association. It's a nonprofit. We have a little booth off on the side, and we've got a conference coming up in about a month in Duluth. So if you're interested in that, if you're going to be in the area, I'd look forward to seeing you up there as well. Let's see here. I'm going to present findings from a research project we did a little while ago. And this was a study looking at different species and how they're going to survive in kind of future climate, different climate change scenarios. And I guess the bottom line is, you know, it doesn't look good for the humans, right? No, actually, that wasn't one of the species that we looked at. So we were looking at vegetation. And the idea is, you know, what do you want on your slopes, on your riverbeds? What's going to survive into the future? And this comes up in a few different projects. I know one story or one project which there was a 3-mile long flood diversion, and I won't name names to, you know, point anybody out, but maybe the description says too much already. But they had put in a bunch of vegetation, and the water conditions changed on them. It was a lot wetter, and all the vegetation died, and they had chronic erosion problems after that. And so that's really the sort of thing that we're thinking about in this. If you make a choice on vegetation and you choose wrong, what could happen to you in the long run? And so that's really the question, or some of the things that we tried to think about with this study. So if you have a project like this one in this picture here, this is Mission Creek up in the Duluth area. It was restoration, and what do you want on these slopes? And that's really the question that we're trying to answer here, is what will a change in climate do to your vegetation? So for this, I'll explain the study in detail so that you really understand the limitations of it. This was done at the St. Anthony Falls lab. What we did is in the summer of 2010, we grew in these microcosms, let's try this one, about 8 different species. And we had 4 different basins that we constructed to plant these species. And the idea was that we'd let all of these different plants commingle, compete, and that way we'd get a better understanding of how they survive. But then in each of the basins, we're going to do something different with the conditions that they're subjected to. And once that's all done, we'll kind of watch and monitor them throughout the year, and we'd harvest them and collect the above ground biomass to get some hard numbers on that. So a cross-section of those basins that you saw in the picture looks something like this. We have walls on the outside. On the inside, which wasn't really apparent from that last photo, it's a sloping bed. So it really simulates a kind of a riverbank or a shoreline area. The water, we have some plumbing in here. So if you're familiar with the St. Anthony Falls lab, we do draw water out of the Mississippi River, and that was routed through here. We have different ways to control the water level. And then on the sloping bed, we put a bunch of flags, and we put a plant at each one of those and kind of had our network that way to document what was in there. The species we used in the study for the common names, we had 4 species that were more of a wet sort of a species, maybe below water, and 4 of them maybe kind of above the water line, a drier species. Fox sedge, bottlebrush sedge, common rush, prairie cordgrass were the drier ones, and then the giant burreed, river bulrush, sauce-stem bulrush, and lake sedge were the wetter ones. As you heard in my bio, I'm not a biologist. I don't really speak Latin. If you are, this is what we're using there with the wetter and the drier species, with the common names in there as well. And so you can see we did grow this outside. It's kind of in the shadow of the St. Anthony Falls Stream Lab. It's in the lesser known outer spillway, kind of living in the shadow of its big brother there, the famous brother. And so these basins became very jungle-like fairly quickly. We had good growth. But within this, you can actually still see, if we zoom in, where you can still see that the plants did, they were planted. We planted 6-inch pots at these designated flags, as I'd mentioned earlier. Some of them kind of behaved, they stayed put. You can see this picture of the common rush. And in this basin, it is fairly flooded as well. You can see this is actually the high end of the basin, and we have water all the way up to the full end. So this is kind of a fully flooded basin. There's other species in here. This is the bottlebrush sedge. We also have the river bulrush and the soft stem bulrush here, this more tubular one. So you can kind of pick these out in the jungle. And that's a big part of what the project did, was going back to document this and look at this. So in cross-section, just to remind you again, so we have all these different species, and we have these water levels that we really control. So that's one of the limitations, I guess, on this when we talk about a climate change scenario. We were really just looking at inundation depth. We weren't able to, or we didn't have the budget to, tent over these and really control other factors that we might see in a future climate, as far as different solar radiation or different humidity, temperature, these sorts of things. So it was really just looking at the inundation levels. And so we flooded out some of these, and these other 4 basins received lower water level depths. So those were determined, that amount of inundation and kind of the pattern of that inundation, that was based on fairly significant climate change modeling, and that was done by Sharon Masagi and Camilla Carell. So they'd done a lot of thought and modeling to come up with these plots where we basically have a dry basin, we have a wet basin, we have what's a normal basin, and we also tried to develop a fluctuating basin, which is really, gives us a bracket of different climate futures with these plant communities. So, we kind of did what we could with this as far as making the best with what our budget would allow. We chose 8 species that are maybe common restoration species. We chose, kind of bracketing water conditions. But we still have a lot of limitations with this, like I said. We don't really know what the temperatures and how these other aspects would impact these communities. So that's kind of the start of it, what we put together and what we monitored for a season. Came back at the end, harvested everything out, weighed it, kind of checked everything. And this was all the plant identification that was really done by Karen Terry and Mary Blickendorfer. They were really the plant experts that could really identify this stuff. I can't identify a plant for my life depended on it, really. But with all that, with pushing these limits on what we had, the statistics got kind of complicated as is. So I think we pushed it about as far as we could. Now, in these basins, this is one way to look at the results. If you can see the values in each of these boxes, then your eyes are pretty good. What you should be looking at, though, is just the colors that we have here. These the darker cells are where we actually planted one of the plants originally. This box represents one of those basins, one of those microcosms. We had 16 different columns of plants that went in, and we had 14 rows going across. So the river bulrush species in the wet basin was planted where these darker shaded boxes are. And then at the end of the season, when we went back and basically indexed these and harvested them, we noted where the plant had now migrated to as well. So you can see we have a fair amount of migration right through the middle of this, where even though they weren't planted, they were only planted to about this middle line, they started to migrate upwards into the basin. all of these empty boxes had a different species planted in them. So the river bulrush was just put in these one set of boxes. And so it was basically out-competing or spreading a little bit there. In the dry condition, interestingly enough, it spread everywhere below kind of the halfway point to the cell. So all of this area up here was very dry. The water was fairly low in this basin. On the fluctuating condition, it pretty much went everywhere. This was a fairly aggressive species. And in the normal condition, it was very aggressive as well. So this is one way to look at this species. We can contrast that with lake sedge, which didn't really do anything. Again, all of these shaded, green shaded cells are where that was planted originally in the wet basin, the dry basin fluctuating, normal. We had a little bit of spreading in the fluctuating. In the wet basin, the red cells actually indicate where it was no longer present, so it actually died off, and we lost those. So much higher mortality rate, much less spreading with the lake sedge. Another way to look at this, like I said, we harvested the above ground biomass. So instead of just looking at where we saw the plants, we can get similar plots looking at this above ground biomass value, and you see I have all 8 species, so this gets rather difficult to see, but similar trends to what we saw. Some species maybe have a lot of growth in the wet end of the basin, so if you line these up, this would be the wet end going up to the dry end, and it tapers off on some of these species. Some of the species that did not do well have very low biomass and don't seem to change much throughout the basin or the from climate change to climate change, or climate scenario to climate scenario. But this plot maybe cleans that up a little bit for us to look at this kind of a histogram. So I've summarized the species down here: fox sedge, bottlebrush sedge, common rush, prairie cordgrass, giant burr reed, river bulrush, soft stem bulrush, and lake sedge. And I think what's obvious on this plot is you can see we had 2 species that really dominated in the biomass. And we saw one of those earlier with the river bulrush. It was aggressive, it spread throughout the basin, really outcompeted a lot of these other species. But within that, you can see the wet basin to these others, there was still a fair amount of variation. Other species, we saw the lake sedge earlier, really did not have a very high biomass. And we saw, that was fairly consistent across all the basins as well. So with the statistics on this, it has to be done with a split plot with nesting in rows. And so if you look at it more as an inundation within an individual species, like river bulrush, we can plot the data up this way. And we're looking for statistical differences between the rows in each of the basins. Bottom line is I've circled the, remote kind of jumps on you. We'll leave it there. The circled spots are where we did see a statistically significant difference within that species. So you can see the river bulrush, generated a lot of biomass. And one question would be, with different climate scenarios, is there statistical difference from basin to basin? And for river bulrush, we did see that. We saw statistical significance on several of these different climate scenarios, 3 of them. Where another species like prairie cordgrass or common rush here, you don't see any statistical difference. So even though some of these look fairly broad, you could say that maybe our experiment couldn't see any difference between these basins. So that's information for us as well, to think that some of these species are maybe a little bit more kind of middle of the road. They will survive in a variety of situations or have a similar response in a variety of situations. And so that gets into our biodiversity as well. We saw 2 species that really dominated this system heavily. And that caused our condition of diversity changes. And so if we do a Shannon-Wiener index on diversity, the wet basin had the least diverse of all of our situations. And our normal condition actually had the most diverse. But it wasn't too much different from the fluctuating. So we do have to be sensitive to that or consider that when we look at these different species. So why did we see these changes? I think that is a really good question to ask. And if you're going to use these specific species, this might be of value to you. But if you're not using these species, what can we infer from kind of the bigger picture on this? And I think it seems like it's related to the rhizomatous nature of the roots, where it allowed for some of the species, like the river bulrush, to migrate and spread and basically move to where it was best able to compete in that water environment. So I think that was one thing that we suspect kind of drives a lot of this. We can look at other recommendations specific to these species. The common rush, the prairie cordgrass, the giant burrweed, all seem to grow well, but we do have to be a little bit careful on the giant burrweed that it's not too aggressive and may result in a monoculture in the end. Fox sedge, bottlebrush sedge, lake sedge, not real strong growers. I think if you're going to include them in one of these communities, you may have those dying off over time or really not thriving very well. And the river bulrush looked like it was a very kind of diverse, it was a strong grower. But again, it could result in monoculture. It might be possible to pair a couple of these stronger ones together, like the giant burrweed, the river bulrush, to give us a little bit of diversity. If you have a situation where you don't know or you suspect there's going to be a highly variable future climate condition, pairing a couple of these species might be a way to kind of hedge your bets, get a little bit of diversity, still provide some erosion control. They have some aggressive nature to move around a little bit and kind of adapt to the conditions. And I'll point out that we do have a paper out on ecological restoration that has a lot of these findings if you want more of the details. There's also a 319 report that was our funding for this. Barb Luukkonen wrote that up, so that'll have more details as well. If you want more info on these, I should thank our funding sources and all of the people that helped to pull this project together as well. We did have a lot of people involved with the counting and the measuring and all of that. I think I'll end with these numbers. 30,000, 10,000, and 10. It's actually days, minutes, and seconds. We've got 30,000 days. It's about an average of what we all have. 10,000 hours is what it takes to get really good at something like river restoration. And so that's kind of what we have to work with to make a difference. 10 seconds. It's an average attention span. That's what we're fighting against. So keep that in mind as we're going up there. I guess the last one, maybe a little hope. 8 seconds, my understanding, that's the attention span of a fish. So we could be worse. All right. So I think we're done. Any questions?
Does anyone have any questions for John?
In Iowa and some other places, a lot of the vegetation sustainability, realizing the duration of the flooding, and there's been some years that we had a really wet period, that you had inundations lasting sometimes from 3 days to 10 days to even over a month. Have you included some of those long duration periods to see the survivability of these or other species than the ones that you've done in this study?
That is a great question, yeah. So, and that is kind of the basis of our research, was looking at these durations. And we tried to get, kind of bracket these different durations where maybe the dry has a the shortest duration, and the fluctuating had different durations. So we haven't looked at any other species besides these at this point. And I think that there is a lot more that could be done. I think it's unfortunate that this research is specific to maybe this community, this environment, this plant selection, and understanding how it might behave in other situations. That's more research, more studies that really need to be done. So I don't have any other species that I can give you insight to at this time.
We have time for one more question. Anyone else?
Fair enough question. I was wondering, I think you kind of answered it already, more studies, more everything else, but on substrate type. If you looked at different substrate type, spreading there, and whatnot.
Right, yeah, so the question is, various different soil types and how would that affect it? And that is a great question. For this study, we did use for uniformity across all the basins. We basically imported engineered soil so that we knew they were all consistent and mixed well and that sort of stuff. So it was a mix of sand with a little compost in it to try to get kind of a uniform growing thing. And that's definitely more variability, more things that could be looked at with how it would survive in a different soil type. Absolutely, yeah.
Let's thank John.